Welcome to our cell and molecular biology lecture. Today, we are going to explore a fascinating topic called RNA editing. Imagine if the genetic code you inherited was not a strict rulebook, but rather a rough draft that your cells could rewrite on the fly. This incredible ability allows organisms to adapt and create immense complexity from a limited set of genes. Let us dive in and see how this works. To understand what RNA editing is, let us start with a simple analogy. Imagine you are an author who has written a brilliant book. The original master manuscript is locked safely in a vault. In our cells, this vault is the nucleus, and the manuscript is your DNA. When you want to print copies, you do not risk the original. You make a photocopy. This photocopy represents the messenger RNA. But wait, before the factory actually prints the book to make the final protein, a special editor comes in with a red pen and changes a few specific letters on your photocopy. This means the final printed book will have a slightly different story than the original master manuscript. Now that we have our analogy, let us look at the formal biological definition. RNA editing is a specific type of post-transcriptional modification. This means the actual nucleotide sequence of an RNA transcript is altered after it has already been synthesized by RNA polymerase. Unlike RNA splicing, which removes large chunks of the sequence, known as introns, RNA editing involves very small, precise changes. These can be inserting, deleting, or chemically modifying individual RNA bases. In mammals, the most common types of RNA editing involve base substitutions. Specifically, we see the chemical deamination of cytosine to uridine, or adenosine to inosine. Because the ribosome reads the messenger RNA sequence letter by letter, changing even a single letter can change the amino acid that gets added to the protein chain. This brings us to an important question. Why does RNA editing matter so much to living organisms? The answer lies in efficiency and evolution. RNA editing is a brilliant evolutionary strategy to maximize the number of different proteins a cell can make without needing to increase the size of its DNA genome. If a cell needed a separate gene for every single slight variation of a protein, our genetic code would become incredibly bulky and much harder to manage. By using RNA editing, the cell can use a single gene to serve completely different functions in different organs or at different stages of development. It is a highly elegant way to generate biological complexity from a limited set of genetic instructions. Let us see this brilliant strategy in action with a real biological example. In the human body, there is a gene that produces a protein called apolipoprotein B. In the liver, the messenger RNA is left completely unedited. This produces a massive protein that is perfectly designed to transport cholesterol in the blood. However, in the intestine, the exact same messenger RNA transcript encounters a specific enzyme. This enzyme edits a single cytosine base into a uridine base. This tiny change creates an early stop signal during translation. As a result, the intestine produces a much shorter version of the protein, which is specialized for absorbing fats from your diet. One gene, two completely different functional proteins. To truly grasp this, let us look at a diagram of the apolipoprotein B editing process. On the left side of the screen, you see the unedited messenger RNA strand containing the sequence CAA. When translated, this forms the large, long protein chain labeled ApoB100, used in the liver. Now, follow the arrow through the pink enzyme, cytidine deaminase. This enzyme performs the edit, changing the C to a U. Look at the edited messenger RNA on the right. The sequence is now UAA, which is a stop codon. Because of this stop signal, translation halts early, resulting in the much shorter protein labeled ApoB48 used in the intestine. This visual perfectly captures how one tiny edit drastically changes the physical output. Now that we understand the biological mechanism and its visual impact, we must ask how scientists actually study this in the lab. While RNA editing is a biological process, researchers often need to measure exactly how actively it is happening in a given cell. They do this by calculating something called editing efficiency. This metric helps us understand exactly what fraction of the genetic messages have been altered by the cell's editing enzymes. By quantifying this, researchers can compare editing activity between healthy cells and diseased cells, which is crucial for understanding certain genetic disorders. If the editing efficiency is high, it means the cell is actively modifying a very large portion of its genetic messages. To measure this activity, we use a straightforward mathematical formula. Conceptually, we just want to find the percentage of transcripts that got edited. The formula is E equals N subscript E divided by N subscript T, all multiplied by 100. Let us break down these symbols. E represents the editing efficiency, which is always expressed as a percentage. N subscript E is the number of RNA transcripts in our sample that have been successfully altered by the cellular enzymes. N subscript T represents the entire pool of that specific RNA transcript present in the sample, including both the edited and the unedited versions. Physically, if the number of edited transcripts increases while the total pool remains the same, the overall efficiency E will increase. Let us apply this formula to a real research scenario. A researcher is studying messenger RNA transcripts for a specific neurotransmitter receptor in a mouse brain. We are given that they extract a total of 5,000 transcripts. Upon sequencing, they find that 3,250 of these have undergone an adenosine to inosine edit. We need to find the editing efficiency. We choose our efficiency formula because it directly compares the edited portion to the whole. We substitute 3,250 for N subscript E in the numerator and 5,000 for N subscript T in the denominator. Dividing these gives us a decimal of 0.65. Multiplying by 100 gives our final answer of 65%. Physically, this means the majority of the receptors produced in this brain region will have the modified structure, which likely changes how the neurons communicate. Now that we can calculate efficiency for a single site, let us expand our view. What happens when a transcript has more than one editable location? Some messenger RNA transcripts have multiple spots where editing can occur, creating even more variety. The cellular enzymes edit these sites completely independently of one another. You can think of each editing site exactly like a binary switch in a computer. It can either be in an edited state or an unedited state. Because these sites are independent, the combinations multiply. This means that adding just a few editing sites creates an exponential increase in the functional diversity of the proteins a cell can produce from just one single gene. To figure out exactly how many variations are possible, we use a simple exponential formula derived from the fundamental counting principle in probability. The formula is V equals 2 to the power of n. Let us define these terms. V represents the total number of unique protein variants that can be produced from the gene. The variable n represents the number of independent editing sites present on the messenger RNA transcript. The base of the exponent is 2 because, as we just discussed, each individual site has exactly 2 possible states. It is either edited or it is unedited. Physically, as the number of editing sites n increases, the total number of variants v scales up incredibly quickly. Let us see this exponential growth with a quick example. Suppose a single messenger RNA transcript has 3 distinct locations where an adenosine can be edited to an inosine. We want to know how many unique protein variants can be generated. We are given that n equals 3. We use our diversity formula, v equals 2 to the power of n, because the sites are independent binary choices. We substitute 3 for n, giving us 2 to the power of 3. Calculating 2 times 2 times 2 gives us 8. Our final answer is 8 unique protein variants. Physically, this beautifully illustrates how a single gene can produce 8 completely different protein versions just by mixing and matching edits at 3 locations, requiring absolutely no extra DNA. We have covered a lot of ground today, so let us summarize the key takeaways. First, RNA editing is a powerful post-transcriptional tool that allows cells to rewrite their own genetic messages before they are translated into proteins. Second, we saw how precise single-letter changes can create multiple, functionally distinct proteins from a single gene, like we saw with apolipoprotein B. Third, we learned how to calculate editing efficiency to measure this activity and how multiple editing sites exponentially increase protein diversity. Ultimately, RNA editing proves that the DNA sequence is not always the final word in determining a protein's structure and function. You now have a solid understanding of this incredible biological process.